Hi, my name is Gavin Jordan. Thank you for clicking onto my long talk about cancer and tumor suppressor genes. Now, what I want to get across in this presentation is to elaborate on the complex nature of cancer, the way that cancer originates as a problem for multicellular organisms, how cancer can be the result of a variety of different problems and outside factors, and how cancer has this interplay across species that presents an interesting paradox that has been observed on in the past. And as a result of the complex nature of cancer as an evolutionary problem, the counteraction of tumor suppressor genes in response. So, trying to understand how tumor suppressor genes function, what they're used for, and how we go about actually identifying a specific type of tumor suppressor gene. And for our specific purposes today, Arcan 1.4. And hopefully, at the end of all of this, I want to be able to elaborate on the broader implications of cancer and tumor suppressor genes as they kind of coalesce at the end of what I'm going to be talking about. But to begin with, I want to dive into the complex nature of cancer itself. Now, cancer is a huge field of study. There's so many things to talk about in regards to it, but I think that it can be most basically summarized in this autonomous cell syndrome analogy, this little truncated version of it. Because cancer fundamentally is the lack of response or adherence of a cell or a variety of cells to the signals of cells around it. It develops its own sort of pathway, right? A cell can have its own specific functions, and when the cell is damaged in some way, it can begin to deviate from its intended purpose. It stops listening to the cell signaling of cells that surround it, and it becomes its own autonomous cell that does not want to cooperate with that of others. And it also, most importantly, doesn't want to induce apoptosis and die. It also vehemently wants to avoid the immune system. Now, cancer can have a variety of different origins. It can happen in any cell in the body. In fact, it could be the skin cells, it could be muscle cells, it could be neurons, it could be bone cells, it could be anywhere at any time, as long as the cell is able to replicate and has some mutation in it that causes it to cease functioning in conjunction with the cells surrounding it. And the reason that cancer exists is complex, but the most basic premise of it is that it's an evolutionary problem. It's a consequence of life, especially life such as ourselves, being multicellular. If we are a collection of cells that has to function in conjunction with each other, we have to be able to signal and work together correctly. And sometimes when cells break on replication, it doesn't follow that pathway and it doesn't follow instructions. And it's just going to happen. It happens all of the time. It's just that as our immune systems begin to degrade, it can't function as well as it could in finding these cancerous and malignant cells. And cancer has its own variety of methods to going in and avoiding immune response. So it engages in this sort of cat and mouse with the body's immune system. So the body has always had this pressure, this evolutionary pressure, to evolve a means of dealing with cancer in an effective way. And this figure explains part of it. It's cancer suppression by increasing tumor suppressing genes. And we can see it with suppressing these oncogenes as well as the variety of interplay. But most importantly, I want to talk about tumor suppressor genes as an overall concept. And to begin with that, I want to talk about oncogenes versus tumor suppressor genes. So we've talked about cancer as a broad subject. There are a lot of things that can cause it, but one of those causes is this thing called oncogenes. And these are, the root word oncology, related to cancer. These are these necessary genes that might be involved with replication or a variety of other functions that, when left to their own devices, when left as an overactivated part, can lead to further cancer. Now, we can kind of equate oncogenes to a gas pedal in a car, and the counterplay to that is tumor suppressor genes, which function as the brakes. So the purpose of a tumor suppressor gene is to induce some sort of stopgap in the cell to keep it functioning how we need the cell to function, right? If it's replicating too often or it sees that there's something wrong in the replication machinery, it can induce a replication arrest, and it can say, hold on, we need to repair some DNA, stop replicating, stand by in this stage and wait. Or it can say, we're too far gone, we're pulling the emergency brake, and we're going to induce apoptosis, and that's the destruction of the cell. It decides it's going to practically self-destruct. Now, if tumor suppressor genes were so effective in the first place, well, why is there cancer? Well, there's a variety of ways that cancer can get around tumor suppressor genes. And mostly, when cancer becomes cancer, when something goes wrong, it's when these oncogenes are functioning at a rate higher than the tumor suppressor gene is able to stop. So it's like slamming your foot down, throwing a brick down on the gas pedal, and it starts chugging forward. Even if you have the brakes down, the brakes aren't good enough to stop the car entirely. Another way is if I cut the brake lines and the brakes just don't work anymore, the tumor suppressor gene, through radiation or some other mutagen, gets destroyed and it doesn't function. These oncogenes just keep pressing on the gas pedal and cancer begins to develop rapidly on its own. But so I want to focus on a specific sort of tumor suppressor gene, and that tumor suppressor gene is ARCAN 1.4. And this is a little complicated, but basically ARCAN stands for regulatory calcineurin. It's a regulatory element of calcineurin. But calcineurin is, what is calcineurin? Well, calcineurin plays a role in the immune response as this calcium dependent mediator. When calcium is introduced into the cell for the purposes of inducing an immune response, it comes down here where NFAT, this NFAT, calcineurin NFAT C1 pathway, it functions as this mediator where a phosphorylated NFAT protein is bound to calcineurin, passes through the nuclear membrane, and activates these immune response genes. So we're looking at possible immune response, and ARCAN 1.4, that fits the bill. It's one of these regulatory elements of calcineurin that, when not present, can have a negative side effect in breast carcinoma. And that is where our next study, the study I want to focus on, comes from. It's from Deng et al. From the Journal of Molecular Cancer. And the premise of this study that they performed is that when ARCAN 1.4 is knocked out, it causes breast carcinomas to behave aggressively, aggressive malignancy. So the purpose of this paper and how they really accomplished it, now that they've formed this background to it, We know it at RCAN 1.4, and RCAN specifically is related to this regulation of calcineurin, which leads to immune response. What are we going to do about it? Well, if we think that it's a tumor suppressor, we should go in, knock it out, and see if it has any effect on carcinoma cell line. So the premise behind it is to perform knockouts using CRISPR Cas9 and observe the differences between wild type and these carcinoma cell lines that don't have RCAN 1.4. And to do this, we use CRISPR-Cas9 gene disruption. And the basic premise behind this is the use of plasmids and RNA to guide the plasmids in, which have been edited specifically to move in, target the specific cell or the specific gene, the specific exon, and excise the exon, repair the DNA so that everything functions as it should, simply without this specific exon being included in the genome. And then after that, to test to make sure that this gene disruption has worked, we're doing immunohistochemical assays. These are immunoblot tests. We're going to break everything down. We're going to see if we've produced this type of DNA or this particular type of protein. And then after that, once we've confirmed that we have this specifically targeted, we're going to move on to Transwell migration assays, which is important in cancer because cancer's ability to migrate around, to invade into different spaces that they're not supposed to be in and settle down and start to recruit machinery around it to support themselves. So having high migration and invasion scores is more of an indication of malignancy factors. And once we see, hey, if there's a difference here, we're going to move on to in vivo mouse models and see, once we put all of this stuff together, we know we have the specificity, we know that we have some difference in behavior. What happens when we put it into some live mice and let it go? So our results from this are really interesting, very, very cool. To begin with, the immunoblot test, this is just to confirm that, hey, we have knocked out RCAN 1.4 with our CRISPR-Cas9 interference. And RCAN 1.4 is going to be denoted by these red circles. And 1.1, which is another exon in this RCAN1 gene, we're going to make sure that we have it specifically so that 1.4 is the only thing taken out. And you can see that they accomplished that. Wild type in these various immortalized breast carcinoma cell lines, MDA, MB, and BT549 and HCC 1806, the wild type and these two in figure A are, they have these blots in 1.1 and 1.4, and in RCAN 1.4 knockout 1 and 2, they're missing. Another part of this is GAPDH. You might be wondering about that. It's a specific protein that's involved in apoptosis machinery. So not only are we getting rid of a specific exon from RCAN1, we are also making sure that we're not just deleting apoptosis machinery entirely. We're just looking at this exon. And they accomplished it. In these blots, you can see after the knockout, nothing's here. We've been able to completely knock it out in both cell lines without any collateral damage. CRISPR-Cas9 specifically worked on 1.4. And down in figure B, we have sort of the inverse. Vector is what we're going to be using in our in vivo assay. That's when we're programming it and we have everything available. Versus wild type of varieties, RCAN 1.4 OV6 and 7. Here we're showing that in our vector that we're specifically programming for, 1.4 is gone, 1.1 is still the same, GAPDH still there, and that's the same in the wild type. So that's going to carry on as a motif in the next figure, but hey, we're showing we've knocked it out, we've done it. So Here in figure C, which is going to be our Transwell migration, we've shown we have knocked out 1.4 exon. Let's see what happens when we put these cells that we have RCAM 1.4 knocked out. What will it do in a migration assay? And you can see here up at the top, we have these cell lines again, MDA, MB, 231, etc., wild type, and our knockouts. And I think the most important thing here is just to observe the differences between wild type and the 1.4 knockouts. You can see there is a significantly higher percentage of migration with each of these knockouts that is not there in wild type. And the same is true with the invasion percentage, which is the ability to transgress across a membrane and pretty much destroy it. It's forcibly making its way through a membrane. And you can see it still performs even higher than wild type in these assays. And that's consistent across these cell lines. And on the flip side, like before, HCC 1806, where we have our vector, it performs higher than our wild type varieties at the same job. So consistently, where 1.4 is knocked out, we have a migration percent increase and an invasion increase. So we're seeing, hey, these cells, when we knock them out, they're aggressive. They're going in, they're going across a membrane, they're invading cell spaces that they really shouldn't be, much higher than they would in wild type. So now that we've established that, well, let's look in vivo. And here is where I think everything comes into focus. So this is an in vivo experiment using naked mice where we've taken these carcinoma cell lines, they're already cancerous, and we're seeing what happens when we take out 1.4 versus these carcinoma lines, which are wild type. And one of the important factors, really important, is they're all tagged with luciferase. And luciferase is a protein that allows for light production. And that allows us to do bioluminescence imaging, which is indicated here by these photon flux scores. So this is showing the density of light in these specific areas. And you can see that wild type at the end of these 3 weeks do not have very aggressive tumor locations at the site of injection. There's practically nothing at the end of week 1. Week 2, you have smaller, not as dense injection sites. And by week 21, you're starting to get red at the point of injection, but they're localized at the injection sites. And when you compare that with RCAM 1.4, where you do have these photon flux indicators here at the injection sites at week 1 compared to day 7 where there's nothing in wild type. Day 14 where there's aggressive expansion, plenty of red spots that have spread across the head and across the injection sites into the thoracic and abdominal cavities, as well as joining with other injection sites across the posterior. And then finally, at the end of the 3rd week, where most of the mouse is covered up with some form of luciferase indicating cell. There's a lot of red here, a lot of spread of cancerous and carcinomic cells. There's a crazy indication and great stark indications between these two types of mice, wild type and 1.4. And we can even see down here in the graph, just to show us the metastasis burden, which is the normalized photon flux across all of this experiment, you can still see massive difference in the significance from here up to here with our standard deviation. It's really, really indicative of just how much the cell lines are acting differently versus wild type when we have this important calcineurin regulator knocked out. So the conclusion of this, what we've accomplished is RCAM 1.4 by itself functions as a tumor suppressor by being involved in this immune response and apoptosis pathway. We've established that our CRISPR-Cas9 disruption worked. We came in, we focused on specifically this exon for this machinery, and we took it out. And once we did that, carcinoma cells began to migrate and invade much more easily. And ultimately, they became aggressively malignant and able to migrate in this in vivo naked mouse experiment. So that was a great study by Dang et al. Very, very cool results. But the ultimate take home that I want to get across with this study and talking about cancer and tumor suppressor is that cancer is a very complex side effect of multicellularity. It's the result of having to have a lot of different cells working together to accomplish a higher goal, and things go wrong all of the time. And this sort of uh, cancer in general is kind of paradoxical across species. It's not just affected by the amount of cells in a body. It's not like blue whales are dropping dead of cancer all the time, even though they're the largest animal on Earth. They have evolved to meet these pressures with the tumor suppressor genes as an evolutionary pressure, just to say, hey, we know this is a problem. It can totally kill us. We need to have a stopgap to control our cells to keep them from going off the rails. And when we lose these tumor suppressor genes, it can definitely lead to cancer. When they don't have that brake to their gas pedal, they can become malignant, and we can see the propagation of cancer as an aggressively malignant sort of cell line, like we saw in the study that we just witnessed. So ultimately, knowing the variety of tumor cell genes and tumor suppressors is important to us, because that's how we can characterize cancers, because cancers can be specific if they're very specialized. It could be the result of oncogenes being overexpressed. It can be the result of tumor suppression genes being damaged and not working entirely. So knowing what the tumor suppressor genes are and how they function in respect to transcription factors and a whole bunch of other factors with the immune system is important for us to understand specific types of cancer in different patients. And hopefully, when we gain the ability to more easily focus on immune response and immunotherapy for cancer, maybe we can find some sort of intervention with tumor suppressor genes to reactivate them or reintroduce them into these cancer cells. But that's the hope. Thank you, everybody, for listening to my long talk on cancer and tumor suppressor genes. I very much appreciate it. I hope everybody has a great day. Thank you so much for watching.